Presentation it goes from basic to wonky. So I hope you can bear with me, and please you can ask questions. I have a lot of slides, so we'll see if we can get through them all. But just the general idea is what can be put in the playbook, and how do we want to tackle some of these issues again regarding sample planning and analysis, data analysis planning. So we all know this, microplastics are a complicated environmental target. Interestingly, we talk about non-standard sampling approaches, non-standard methods, lack of systemization and data analysis, and of course, error. I'm going to talk about error primarily. But interestingly, the wide variety of study goals is maybe a big challenge for quality assurance, especially. So as microplastics become more and more important to agencies that regulate or are regulated, thinking of sanitation districts in particular, getting the study goals more standardized is going to make a big impact, I think. And so I'm going to talk a little bit about that. Obviously, we know these different areas where there's a lot of variation generated: a sampling approach, preparation, identification. You guys know all that. I like this little cartoon, and I'm going to use this cartoon throughout the presentation. So we know variability in measurements and environmental sampling causes problems, and we need to use techniques if we want to compare. This is especially regarding quantitative studies. So if we're comparing to a threshold or comparing essential tendencies of two groups, we know we have a lot of variation. And in my career, I've focused on sampling variation primarily, and I'm going to use these cartoons throughout. So there's sampling variation here, and it's got this very uncharacteristic bell curve at the bottom, because we know that this is usually log normal or no distribution with regard to the sampling distribution. The true population may be normally distributed, but with regard to sampling, we don't always get all the samples we need, and so we're going to have some variation on that. Interestingly, these other areas, they may be a little more realistic, especially analysis. We know we can get pretty good repeatability in measurements, whether they be FTIR peak matching or whether they're pyrolysis GCMS kind of things. So I've always in my career relied on the analysis people to do their job, and I focused on sampling and the sampling error in my career. And more in microplastics, I've branched into these other three areas. So this presentation is going to follow these first three boxes. I'm not really going to touch on analysis because that's really Shelley and Wynn's expertise, and they have a lot of great things to say about that. But I'm going to talk about these other three because they're important in sample planning. And when I'm reviewing a sample plan for a partner, or I'm working on a pack, say with Shelley Walter and the WRF, these are the things I'm thinking about especially. So if you take a look at it in this way, this is a classic EPA rubric. How are we comparing our sampling distribution to a threshold? And how likely are we to get the true state of nature that true distribution of contamination? So in this case, the dark line is an unbiased true state of nature, and the dotted line being a biased one. We got a sample, and this is what we saw. In this particular case, we're saying, hey, this site is looking like it's a non-attainment, but it actually might truly be an attainment, meaning under a threshold. And so it really takes a lot of planning to make sure you have adequate samples and good quality measurement for this kind of decision. And so throughout the presentation, I'm going to talk about, microplastics analysis with this in mind, either with estimating, goal of estimating a concentration, or a goal of comparing concentrations. I'm not really going to talk about microplastics for other types of research, microplastics analysis. So bear that in mind. These rules don't apply to everything, but they're really important when it comes to repeatability and reproducibility of numbers. And I broke them up into this fashion again. The precision is up to the quality control procedures, subsampling, sample prep, and then, of course, analysis and peak matching. The accuracy and representativeness, that goes to study goals, conceptual model, and selecting the right analytical, deciding whether it's an estimation problem or a decision problem. So this is kind of my bread and butter, this baseline condition, null hypothesis stuff. It's my favorite topic. I used to teach a class at it at the University of San Francisco, and I've taught all over EPA as well. So here's some of my favorite general study plan references, and I added the Water Board and the new state accreditation program. I'm not as familiar with that, although I have read the guidance. I didn't put enough time in this case into reviewing that, so we'll have to put a pin in that for now. But you can see a lot of other things. I'm going to talk a lot about the EPA quality program. I work a lot with USGS and NIOSH on their different methodologies. I'm a person who does sample planning for all kinds of analysis, all sorts of different stuff, because I'm in Superfund, so we handle a lot of different kinds of things. So I'm looking at this really holistically. Here are all the EPA quality assurance guidelines. So they seem a little boring to have very bland titles. Interestingly, there's three that are very, very useful that I think we can capitalize on for the playbook. And those in particular are G4, which is the use of data quality objectives, which I'm going to talk about. G8, which is really about quality control and how to do your lab program. And G9, which is the data analysis planning, picking what statistics you want to use, whether you have enough samples, those kinds of things. So I'm going to touch on aspects of those, and I think those would be really useful for this playbook, where we talk about those general things, and then we can get into specifics, like for example, that Water Board guidance on really how to do data validation and verification for count-based measurements. Okay, so again, there's my little cartoon. This is the world of sample planning, and what does EPA's guidance say about that, and how can that be applied to microplastics? So I use this all the time, this particular little rubric. And I think it originally comes from the standard methods for water and wastewater examination, if you're familiar, this kind of concept of what's your study goal, what's your conceptual model. You guys will recognize these things. What's your analytical method going to be? And what type of information do you need from that analytical method? Is it going to be a quantitative study or a research study? What kind of thing are you trying to answer? The boundary statement, the time and space that you want this result to apply to. An analytical approach, meaning a decision rule. Are you going to estimate? Are you going to try to compare? So that's not which methodology you're using. This is what's your approach. And the desired level of confidence in the results, that's the p-value maybe, but also the hypothesis that you're testing. What's the baseline versus the null hypothesis? And then the sampling plan itself. And so we use these 7 steps all the time for all sorts of emerging, sampling issues, all sorts of data interpretation. We get called by the news. What's this data mean? I kind of use this rubric to analyze that and come up with the best kind of comment I can make. But in addition, every EPA or EPA-funded sampling plan has to follow these rules. And interestingly, there's a unified quality program among all federal agencies that follow these rules as well. And so there's all sorts of things out there, worksheets and software you can use to fill this stuff out. So I really want to share that with people. That's been a big emphasis. And again, it's just got to do with sampling primarily, about quality assurance, not so much the things that happen here day to day to really make the lab get the best result it possibly can. So here's some examples of study teams, right? At EPA, we make an emphasis on who's on the study team. And so here's just a couple of jokes from my different projects. This one in the ballroom, interestingly, that's Huntington Beach oil spill. So we had Cal EPA OEHA there. We had Orange County Public Health there. We had Coast Guard. We had the Water Board. We had EPA. And we were tasked with figuring out how to evaluate whether there was oil contamination in sand or beach water. So we had to bring all those people into the room and try to figure out a game plan of what we wanted to sample and where and how we wanted to interpret the results kind of on the fly. So this is the kind of experience I have. In that other picture, we're in a command post doing air surveillance after a big fire. So that kind of experience has trained me in this approach. And this is very familiar to you guys. You guys see site conceptual models all the time. You see them in big reports, and if you're lucky, you'll see one in a scientific paper. Meaning, what is the link between your bigger understanding of this problem and your study design specifically? So what is that link? How do you make that jump from the cartoon in the presentation or in the guidance document to something that you can test with environmental measurements? That's a big challenge, and we take it for granted. So it's nice to actually make these cartoons and put it in there. So cheers to these guys for doing that. And then to go another step further, how do you go from that conceptual cartoon to a sampling map, like where the samples are actually going to be and how many? So these are cartoons from the early 90s, but they're really important. I use a software now. I plug it in and make a base map, and it'll cast the samples out for me. But they're really important, especially when you realize, oh, that's a lot of samples I need to do to achieve my goal. If I want to compare, pre versus post wetland treatment to see if there's removal of microplastics, requires a lot of samples because that variability just in the sampling alone is so great. So this is an opportunity to remind yourself about that. Here are some rules of thumb that are given for number of samples. So for an estimation study, you can get away with less than 30. Like, here's what I think the mean is going to be, or what I think the median would be for this population in this world, this particular study environment. And then for decision-based, if you're trying to do a t-test, especially, or an ANOVA, you need a lot of samples. So USGS is recommending more than 30 samples be used for anything like that. And interestingly, you can use a power calculator. This is little n for your sampling number. And you can use this ratio of the variance over what we call the gray area. The gray area, if you can see my cursor, that's the acceptable difference where you're willing to, you're willing to be wrong, you're willing to fall on one side or the other of your hypothesis if you're in that gray area. And this T95, this happens to be the one for the t-test. That's for a 0.05 significance. So any of those three numbers in the ratio can change to give you a more reasonable sample number. Or you can, of course, work backwards. This is my sample number, and this is what I expect to have as a p-value or a variance or a gray area, meaning the zone where I'm willing to be off. Shelley, Walter, you have your hand up.
Yes, thank you, Harry. And I really appreciate all of this. And just to clarify with the number, this 30 and greater or less than 30, you're talking about each treatment, not the overall number of samples, correct?
Well, only if you're doing a decision between two groups of some type, right? If you really want to know, again, with a small margin of error, right? Small margin of error, if you care about like 5 or 10 units as your margin of error, right? That's going to be a lot of samples. If you care about 100 units for 1,000, it's less samples. You see what I mean? For the same power. And these are just rules of thumb based on, the distributions of t-test, et cetera. And by the way, you can do all sorts of things to adjust this number. You can take the logs of everything. You can do all sorts of things. You can divide by the volume, all these things to normalize data. Those things also work, and they lower the variance, and so they lower the number of samples you need.
But let's say in a scenario where you have maybe 3 different treatments, and you're looking to compare between those treatments, we're talking about in general, just, just for the scenario, 30 per treatment, not 30 total,
Well,
not 10, 10, 10.
again, it depends on the goals of the study, but
Right.
classically, USGS would say there, yes, yeah, they would. Classically, right? So textbook.
with-
That's not realistic, right? That's not the real world. It's just a reminder that this number has an implication for the variance, for the margin of error, the gray area, and for the p-value itself of the study. And more importantly, it has a bearing on the distribution analysis you're going to pick for your sample distribution, and the match of that back to the true population.
Thank you.
Yeah, yeah. So don't get too worried. I'm not going to make everybody do at least 30 samples for every treatment.
No, I'm not. It's not where I just wanted to clarify.
Yes, yes.
We're talking about is ideal general kind of, it is the heavier number per treatment, not overall.
Correct.
If you have a large study, you're not talking about 30 samples total.
Yes, yes. Theoretically, that's correct. Yeah.
Right, okay,
so just truth be told, I rarely take 30 samples per treatment for a t-test.
right.
Or even one with log transforms and non-parametric. Yeah. But
Sure.
Just the same, that is kind of the general rule that you're taught as you study this field. And so the other way to do it is, okay, I can't really feel good about, I can do this, but I know it's weak, this comparison. But what I can do with less than 30 samples and what's recommended is using the distribution to choose a good upper prediction or upper confidence limit on what you think the mean is. In other words, make it an estimation-based study. You know what I mean? Not everything has to be t-test. You can do an estimation-based. In other words, for these considerations and this plan I looked at, I really feel like this is a strong number. And matter of fact, when people do that, I really appreciate it, because that's a really nice kind of thing to do for representativeness, right? So if I went out and collected samples here in, the outfall of the river, and I did that, I took only 10 samples, I would get maybe an upper estimate of what I thought it would be. That would be my bet for Shelley then to go and do another sample round and see what she got, and we could compare with the same background, the same stuff, and we'd be able to say, yeah, hey, this was a good bet. He bet 95% chance that Shelley would be under this number, or the same, and that's what she turned up with. Otherwise, she found something interesting. The 5% chance there was a change. So all this is analogous to, when you're using an upper confidence limit, you're basically doing a t-test as well, or its equivalent, like normal or non-parametric. So it's kind of interesting when you think about sample analysis planning. So analytical approach, I was talking about this earlier. It's really important to decide between those two types of studies. I think that's really worth doing. Not everything has to be a comparison, to a threshold or to another area. It can be just, I studied this and this is what I saw. And more in the non-academic, where there's no pressure to publish, they're going to be doing that, estimating and trying to show whether they're above or below a threshold, and trying to gauge what's happening over time and try to understand effluent at a water plant, for example. So I think that's a great value, and I think more and more clients are going to want to do that. Here's some other things that we don't really need to talk too much about it, except for I want to emphasize, that people consider the distribution analysis of the data and actually graph it and see what it looks like. See if you have enough samples to satisfy your curiosity about that question. Here's an example. So this is the data from that SFEI study that Diana worked on. She was graciously let me share it. And so just a simple graph, and you can see 8 or 9 samples in these, and maybe that was enough to differentiate between these two populations. So there's clearly not 30 in each of these, but you can kind of see where things are and generally how they're distributed, and these happen to be normalized. They're divided out by the volume, so that also sort of normalizes the data. You can get away with less because you have a smaller variance. But you can kind of see almost a signal here. Like this would probably, demonstrate a significant difference if you did a statistical test. But just kind of interesting to see that. And you can also look at it and say, hey, I think there's some sample points missing. There's some lower ones in this lower quartile. There aren't any. It's kind of odd. And up here, is this an outlier? Should more be collected in there? So I look at this as almost like a fence post without all the fence posts, a fence without all the posts, right? What's missing, what's really going on? Are there certain ranges there more or less? So I recommend this interquantile plot, especially that's called the QQ plot. So that's really worth doing to see what the group looks like together. If we had grouped these together, they would look pretty strange. Certainly log normal, all the data would be pushed to the bottom. And I think that's also an appropriate thing to do, but interestingly, there may be a difference there. Just to think about that. Okay, so that's the sampling portion. So here's my cartoon again, sample prep and analysis, which I'm not really going to talk about, but sample prep is here. And this is kind of an interesting question of, if you're doing sample prep, we have an ASTM for this. Obviously, there's error that's generated because you're not always going to get the same result. So there are ways to do that, and there are ways to evaluate it. I put some over here. The relative percent difference is really common. Relative standard deviation is a nice way to do it. It's the ratio of the standard deviation and the mean that you've got from your results. And then the relative standard error is an interesting one. That's the weighted relative standard deviation. So it's weighted for the different ranges within the data, low, medium, and high. So those are all really interesting, and you see those in the literature and in guidances quite a lot. And you want to think about those. It's just our plug for ASTM. We have a sample prep method. We have a matrix spike method. And we have some sample collection methods. And finally, now there's a pyrolysis GCMS analytical method that was published. There's an FTIR method that Scott has worked on in draft. I don't know the status of that, though. That's being worked on. Okay, so we've talked about the quality assurance and the sample planning piece. Now, if you go, part of that study team is really to get with your laboratory person, Wynn or Shelley, and say, hey, this is what I want to do for my study. Is how many samples I'm going to have. I did my homework, I promise, and here's what I'm trying to achieve. And then they're going to come back to you with, a result and some precision around that. That's really the quality control piece, whereas the other is the quality assurance piece, in my mind. The accuracy piece is to try to capture the environment, represent the environment. The control piece is the precision piece. That's the way I look at it. I'm not sure if that's classical or not, but it's just, in my mind, it's an easy way to break it up. And so you can see, what precision is. It's the, the ability of these guys in this great lab to, get the same result when challenged the same way. There's repeatability, within the sample, reproducibility between, it's also between labs and other analyzers, perhaps. We've been trying to use relative standard deviation, and this is common. You see in the literature, somebody may say, well, I did this big study and I did a couple of duplicates and I did the RPD, RSD between those two. That's the most common, I think, quality control measurement I've seen in the literature. But yeah, we have them for both mass-based and count-based. And interestingly, a couple of subsampling or discussions, right, that you can see. You've heard, and probably some of you published on subsampling and how many particles you have to count, or how many aliquots of sample you have to do for IGCMS in that case. So there's a bunch of different examples. Well, I think what's interesting about subsampling is, I think in general, it's going to follow this kind of, if it's a count-based result, it's going to follow this kind of distribution here, which is the Poisson distribution. This is from that Wagner study. It's counts from lots of different labs with different, with the same standard, right? And the counts were all over the place. But interestingly, they follow this distribution. And counting studies are supposed to follow this distribution based on counting science for its counting methods. We got this from asbestos, but there's a lot of other kinds of counting methods that rely on this known distribution. Again, if you're doing a quality assurance plan and it's a counting plan, you should kind of, I think, understand that your data are likely to be in that Poisson distribution, at least the raw data are. And so what can be done about that? How many samples are needed for that? And interestingly, there's ways to find that out. In fact, the MDA approach from the Water Board guidance, the minimum detectable amount, that's exactly what that is. It's a distribution equation where you go back and plug in the quality you need, and it's going to tell you the number of counts that you're likely to get for that quality. And so interestingly, just by chance, I think, or not by chance, it does follow this pattern. I worked on one myself. It was filter sections with recounts. This study we did back in, actually some of the samples were from here, but it was done out of the EPA's Narragansett lab. Count-based study for microplastics and sediment. And yeah, they used, we're trying to figure out what's the optimum filter size to subsample. And we also saw that the sample counts follow this Poisson distribution. And then we just sort of figured out, did this fit? Interestingly, I read the Fahrenfeld study. I'm on the pack for that. Included this. They did a bootstrap analysis, which I think comes from Chelsea Rockman. Funny enough, the lower end of the bootstrap, all the way up to the higher end, that also followed the Poisson distribution, funny enough, right? They bootstrap the bottom sample. I don't think the bootstrap requirements fit such a small sample. But anyway, the science, I think, points out that the counting error follows the Poisson distribution. So it worked anyway, which I thought was kind of interesting. You can see the bootstrap, I think, is a really interesting concept. So I put it here just because I like to be wonky about this. Your sample distribution is going to mimic your sample, whatever that was. You're just resampling with the same pieces. Okay, so if you have 5 pieces and you resample them over, and it's like rolling a 5-sided dice. Yeah, that's what bootstrapping is, and you get a population of the amount of times each of those numbers came up, but you don't know if that represents the original population or not. So what they did was they analyzed the full sample, all 80 particles, and figured out how many of each polymer with OpenSpec, and they did a great job with that. And then they tried to bootstrap the different sample sizes to see if they matched. And they happened to match, even though the distributions for the small sample sizes are, I think, inadequate, just because of the sample counting statistics. So I thought that was kind of neat. So the RPDs look pretty good anyway. So it's kind of an interesting concept. I happen to be a big bootstrap person. I think it's really interesting to take a small sample size and estimate what the next sampler might see with that technique. But you really do have to have a big range. You have to have a good initial sample to see how things are going to compare. All right. This is just an example of infrared analysis. You guys know, I don't use this method, but they were the first group we worked with at ASTM. But you guys all know about this. And I just use as an example to talk a little bit about RSDs for counting. So I'm going to tackle that really quickly. Here we are again with subsampling. You know, what are some different ways? In our study, this is what we did. We used RSD and predicted Poisson RSD, and we did a fit. We did that with all those different labs. And we're able to see a really interesting fit that follows that statistic. And you can kind of see that in that paper. Oops. Sorry. Yeah, there you go. So it's interesting, again, this Poisson concept is the most likely for the raw counts, not for samples necessarily, but for the raw counts, and a good fit for that. So I just want to keep that in mind, and I think that should be tested more. Interestingly, we got the concept from asbestos. So for the asbestos standard, you're required to do the Poisson RSD. So that's the asbestos science for your count to see if your particular run, your lab did a good job with that and fit the model. So we think that works for microplastics, but obviously there's limitations. Okay, so over to mass. This is what I've been spending my time on more recently. It's nice to have an analysis system that's so common and relatable to other environmental pollutants. So it's really handy. And this little auto sampler here on the top of the pyrolyzer makes it very easy to run a lot of samples. The downside is they're small, right? So they're not very representative. You really got to focus on subsampling. And the other downside is they're destroyed. There's no way to know any characteristics of the particle. But with regard to getting a mass and having a result that's stable and repeatable, it's been pretty cool. So I'll talk now briefly about the RSDs and how to get that really, really tight. And you can see we can build a calibration curve, high, medium, and low, and get results and do this RPD, percent error, right? And then we're able to do the relative standard error for that based on the different weights. So that's a really encouraging model. Okay, I have 2 slides left. So this is just a quick example of, if you're doing a study, how many lab control samples and what I do. So I'm keeping this. If you ask me for review, I might say some things that you may worry about. So just to tell you, it's in the spirit of trying to get good quality control. So with PyGCMS, because it's so easy, we can run a lot of subsamples. Again, they're small, so that's the downside. You can't really get a lot of sample through there, but they're easy to run and they're easy to rerun. So we can run a matrix spike 5 to 7 times in an hour or 2, no problem. With counting, and I was talking about RSDs and RPDs, we think that repeating the counts 5, 6, 7 times, that study of the sediment back east where they looked at the filter size, they counted each of those filter segments 5, 6 times to get a good percent, RSD on those, and then they plotted those. So trying to do recounts is really something worth doing. Similarly with the blanks, it's no problem to do blanks with GCMS and to run them again and again, and then field dupes. So in the playbook, we're going to talk about the different types of blanks, the different types of duplicates, different types of spikes, and relatively how many times they ought to be analyzed, whether it be for mass or count-based. And we'll use that to talk about precision. We'll use that to talk about for mass or count. We'll use that to talk about subsampling. And I think these are kind of the quality control measures that we'll be highlighting. So that's all I have. I hope that this field grows. I think it's exciting to see people doing all kinds of microplastic studies. And personally, I'm really interested in seeing the big institutions take this on, such as county sanitation, anybody with an effluent requirements, anybody doing stormwater monitoring, and looking for ways to systematize and standardize those are really of interest to me. Regret with this talk, I didn't talk enough about the Water Board's guidance on count-based quality control, but we're going to make sure that that's highlighted and discussed in the playbook, and otherwise just kind of present some rubrics for this, even some either software-based or actual form-based ways to tackle some of these questions. Thank you.
All right. Thanks, Harry.
Yeah.
That was great. I appreciate it.
Yeah.
I could listen to you all day talk about this stuff. Let me stop sharing the presentation. I'm gonna ask for time to ask 1 or 2 questions. It looked like there was a hand up. Is there anybody who wants to ask a question, a quick question with Harry? No questions?
Shelley.
If no one else does, I'll ask a question.
Christopher has a question.
Christopher.
Okay,
do.
good. Okay.
Hey, Harry, I've worked with Brian, Terry, Alan, and now Jennifer CU with great detail. I know she's up at Humboldt right now, which is where I'm supposed to be as well. Been working with her on a bunch of sediment stuff. And she mostly works with the chemical end, as you know, and I wanted to know, excuse me, sorry. Oh, sorry. Does some of the actual counts with the different asbestos methods, how, I know there's TEM and then the other method that you used, different light structures. And when I was doing sediment and water chemistry, my big thing was QA/QC as well. And one of the big things that there was a big, huge breakdown when you're doing counts that we ran into was human error, individual error. And I know collecting a subsample within that group, but because of the different particle size, the particle sizes and the particle composition, some will be breaking down sooner than later. And there's so many questions that are going through my head right now I want to ask, but standardized methods through ASTM, and I know that there are so many different methods with some of the chemical analyses. Do you see in the future that standardization will be kind of wrangled in a little bit more than what's been in the past? Because from a physical standpoint, the particles aren't going to go anywhere per se, but with chemicals, it sometimes does. So I was just kind of curious on what your thoughts are for the future.
Yeah, well, I hope that there'll be more standardization, and I don't know which of these methods is going to be the best, especially for subsampling, for count-based, because that's not my expertise. I have just a little, but you know, my colleague Jeff Wagner at California Department of Public Health, who pioneered some of this precision work, he's an asbestos counter, so that's where we came from with that particular analysis, right? And he keeps a control chart for himself and for his lab. What were his results? And they're not always the same, right? Sometimes the counts were not as great, and sometimes they were pretty good. But what he did say, and what the asbestos guidance says for NIOSH, is that's okay. You can still use it for estimation and for quantitation. And of course, we use asbestos quantitation. We do that all day around the country. So to do that, you have to follow some counting rules. And at least for asbestos, those counting rules are established. Microplastics is more complicated. We've got particles.
Yeah, that's something that we're really worried about, being the keeper of the public trust and the keeper of the sovereign lands of the state. That we're getting a lot of questions from individuals, and now with a lot of the legislation that's going into effect in the state, and now with the report to Congress that I've got here that you can't see, for microplastics and microfiber pollution. So these are things that my agency is getting a lot more involved with, and we're trying to come up with some guidance and trying to come up with some standardized ways. And like I said, working with a lot of the EPA Region 9 folks has been great, and want to continue that. And so thanks. I understood most of your presentation. And I think it was actually cool. So thank you very much.
All right, thank you. Yeah, you know, interestingly, I give a pat on the back to the lab folks here.
Oh yeah, Wayne and
that
Shelley.
blind, that blind lab study with the EIR, the automated IR. The automated IR didn't do any better at counting than the human did, and so that's why the counts were done 5, 6, 7 times, and then those RSDs were generated. So it's really a matter of the effort of counting. Whether it's done by a robot or a person is kind of irrelevant.
Yeah, how much coffee have they had?
Yeah.
So, thank you very much. Thanks. Thanks, Shelley.
Yeah. Thanks, everyone. I see a hand up, but I'm gonna actually move on because we're a little behind. So if we have some extra time at the end, I'm sure Harry will be happy to answer some more questions, and I'm sure if you reach out to him, he'll be happy to answer those questions as well. So next up, we have on our agenda another guest speaker, Sarah Balance. She's from CSU San Marcos, and I will let you go ahead and get set up, Sarah.
Perfect. Okay, can you see that?
Yep.
Okay.
Perfect.
Okay, happy Thursday, everybody. I thank you for that great introduction. I also would like to thank the organizing committee for having me here today. Today, I'm going to present a little bit about what I've been doing for, actually, it's quite recent, this project in the snow samples, very preliminary, but today I'm excited to talk about combining submicron spectroscopy techniques, AFMIR and OPTIR, to detect, image, and chemically identify microplastics and nanoplastics in snow samples. So we all know the problem of microplastics and how they can break up in the environment. They go from macroplastics to microplastics, and some even say break down into nanoplastics. And one of the main challenges is that when these particles break down, they have a larger surface area to volume ratio. So they become more of a adsorbent to chemicals and heavy metals. And this makes it a problem for the ingestion with aquatic life and even potentially harmful to humans. So when it comes to the analysis of nanoplastics, there's actually quite a bit of gaps in the research because conventional spectroscopy techniques can typically only go down to 1 to 10 microns. So the two spectroscopy techniques that I'm going to be talking about today is OPTIR, also known as optical photothermal infrared spectroscopy. So this is a really interesting technique. One of our collaborators in Santa Barbara analyzed one of our subsample aliquot drops, and we're able to simultaneously acquire infrared and Raman spectra on particles. I can't particularly go into this in too much detail because I'm not an expert myself, but I can absolutely give you information if you'd like to learn more about this. So the technique that we use in our lab is known as an atomic force microscopy-based infrared spectroscopy technique. And without going into too much time about this, please feel free to ask me a little bit later or reach out to me an email. Essentially what it encompasses is AFMIR, so you're able to look at tiny, tiny particles at nanoscale precision, and then infrared using a quantum cascade laser, you're able to determine the chemical specificity of a particle. And the image that you can see here is a topographical 10 by 10 micron image collected from the AFMIR on 200 nanometer polystyrene spheres. So the samples that we're looking at today is we have a collaborator out in Utah, and she collected snow core samples out in Beaver Mountains Ski Resort in Utah. And she sent us these samples, and two of them were collected in the front of the ski resort, and then, or one was collected in the front of the ski resort, and the other was collected on the other side of the ski resort. I don't have the specific coordinates right now because of conflicting schedules. We're going to meet and talk a little bit more about that and details about potential sources. But right now I'm talking about the preliminary results, which are still really interesting. So when it comes to the sample processing, in total, there were 8 10 microliter drops of snow water were deposited onto a gold substrate. So this equals 80 microliter drop in total, concentrated drop. So once using a brand new clean glass syringe, from the glass vial that the sample was in, you extracted the sample, 10 microliter drop, and then dropped it on a hot plate, approximately 80 degrees Celsius, and that evaporated, and then you would continue to do the same for 80 microliters. And once we had those samples, those were then examined in the OPTIR and the AFMIR. So when it comes to the data imaging and acquisition, before we even looked at any of the environmental samples, we want to make sure that we're setting up and optimizing the AFMIR to a standard. And that's usually used with PMMA. So if the spectra is perfect and looks exactly like PMMA, we're able to move our sample over. And once we have collected spectra, that spectra was then compared to an open, or not open, but a polymer database known as Wiley Know-It-All. And then when it came to the extrapolation of the plastic counts in an 80 microliter drop, and even extrapolated to milliliters, we used MATLAB in order to count the particles that were in an image, all of the particles. And as you can see here, this is just how I extrapolated the total number of particles in an evaporated 80 microliter drop. Okay, so when it comes to the preliminary results of the OPTIR analysis, I wanted to show this image here. So this is really kind of a larger optical image of the drop. And we have a subsample here at 296 microns. And the interesting thing about this technique is that it scans all of the particles, anything that's larger than 500 nanometers, it will pick it up on the analysis and then simultaneously collect infrared and Raman as it does that. So you're left with lots and lots of data in the end to sift through and look at the spectra and compare if it's a natural polymer or a synthetic polymer. So this sample that we looked at prior, you can see in image A is a sampling location collected from the 80 microliter drop of the snow melt. And this is a Beaver Mountain West subsample. And when it scanned through, most of the particles were natural, but two were classified as synthetic, one of which being polydimethylsiloxane, or PDMS, and another being polyester. And PDMS you don't see quite often, but it is interesting to identify in a ski resort sampling location because this is a type of silicone polymer that is often used in ski wax. And polyester, of course, as we know, is a synthetic material that is widely used in indoor and outdoor clothing or even in gear. And you can see here, so the spectra, the blue is the one that was collected using the OPTIR, and then the red is the matching PDMS and the polyester. And I want to note that this peak right here, the 1735, this is a really strong peak that was used in order to identify potential plastics in the AFMIR analysis. So looking at the nano-IR analysis, we were able to do an infrared scan using that specific wavelength. So you can see here in image C that this is a topographical image, very tiny in comparison to the OPTIR one. We're looking at 50 microns by 50 microns. And of course, this can go even smaller, as you can see in the inset map. This tiny little particle that you can see here in the 3 by 3, we were able to, when we scan at 1735 centimeters, anything that has a high peak in this wave number will show up in the map. So then we're able to do point spectral analysis on this, and this tiny particle here showed a signature that is polyester. So that was really interesting to use those these two techniques in combination with each other and see similar particles. And of course, we're looking at the same sample here, Beaver Mountain West. When it comes to the counts and dimensions and the mass concentrations of the plastic for both the AFMIR and the OPTIR, in total, we found that there were 35 particles that were positively classified as plastics, 32 of which were detected in the Beaver Mountain West sample, and 3 were detected in the Beaver Mountain Mid sample. So just to break these down a little bit based on instrument, because as we know, the OPTIR instrument does look at particles that are larger than 500 nanometers. OPTIR identified 8 plastics, 6 of which being PDMS and 2 polyester. And the AFMIR found 24 polyester particles. And these are, of course, in different various sampling locations in the 80 microliter drop. And then the Beaver Mountain mid sample, the OPTIR identified 2 PDMS samples or particles, sorry, and then the AFMIR identified 1. And you can see in this histogram here, the top A and B image, this is a histogram of the area and the volume of the particles that were identified in the AFMIR, and then the area and volume of particles detected in the OPTIR. And similar to lots of publications that have been out about microplastics, we found higher plastic counts with smaller dimensions. So we found a lot of nanoplastic particles using the AFMIR. In fact, 71% of these classified plastics were found to have thicknesses smaller than 1 micron. And those sizes ranged from 14 nanometers to 10 microns. And we're going to go a little bit into that in the next slide about the 14 nanometer particle. And looking at the individual samples using the plastic counts and extrapolating it to 80 microliter drop, we can average approximately 86,000 microplastic nanoplastic particles in that subsample, following approximately 55,000 microplastic nanoplastics in an 80 microliter drop in the Beaver Mountain mid sample. So just a reminder that 80 microliters is quite literally a pin drop. So very, very, very tiny. And then lastly, when it comes to the total mass concentrations, the really interesting thing about the AFMIR is that you're able to determine the length, the width, and the height of the particles that you're identifying. So with that, knowing those the compositions, we're able to determine a mass. So in the one subsample, the Beaver Mountain West, we approximate 171 nanograms per milliliter. And in the other, approximately 145 nanograms per milliliter. So this doesn't seem like much in nanograms, but of course, when it comes to pollution and contaminants in water, of course, a little bit goes a long way when it comes to human health. So this is an image that I really love to show. And this originally came in a 50 by 50 micron. And like the other one, I zoomed in on a very, very tiny particle. And it was originally this one here. So this is a 3 by 3 micron. And similar to the other one, this was run at 1735 centimeters. And the interesting thing is that if you run it at a higher resolution, at a slower scan rate and a smaller size, you're able to pick up particles that you actually didn't see in the 500 micron. And this was the case here. So scanning over it, you can see that these natural particles, you do not see them in the infrared, but you see the bright yellow ones, and these ones came up. And collecting point spectra on this one in particular, we found that this had very similar, if not exactly the same, spectral fingerprint as the larger nanoplastic particle here. So this one here is the smallest one that we identified at 14 nanometers. And of course, this is the detection limit goes down to approximately 6 nanometers pixel size. And you can see that the blue PES signal is from the larger one, and then the 3 here is the red one. And then the black is of the natural particle, and it does not have a distinct signal in the 1735 here. Okay, in conclusion, we find that an 80 microliter drop of snowmelt may contain more than 50,000 plastic particles, estimating 880 million plastics in 1 liter of snow. Of course, that is an extrapolation, concentration. I know that we are slowly moving towards mass-based concentration. So based on the total plastic counts used to extrapolate an 80 microliter drop, we estimate greater than 140 nanograms per milliliter of these micron and submicron plastics in snow. And of these classified particles, 71% of them have thicknesses smaller than 1 micron. So very, very, very tiny. And when it comes to the compositions of the particles that were identified, it really does exemplify the direct influence of humans in the environment, especially when it comes to PDMS. There could be some type of scraping on the dry snow when it comes to the skis and the ski wax. And when it comes to polyester, of course, could be atmospheric transport or just the shedding of any type of fibers. And lastly, while the effects of these nanoplastic particles on human health remain uncertain, there are a lot of gaps in the research, especially when it comes to this type of vibrational spectroscopy research. I don't have a lot to go off of because, of course, it's all trial and error and it's new. Considering the ubiquity of microplastics in total in the environment, further research needs to be conducted in order to better address these potential risks to humans. I just want to say thank you. I would like to give a big shout out to the funding of CSU Coast. Without them, this wouldn't happen. And my boss, Dr. Gerardo Dominguez, and my wonderful collaborators. And I would like to say that my time is coming to an end when it comes to my postdoc, and I wanted to have the opportunity to say that I'm looking for job opportunities and always forming new collaboration. So please feel free to email me. Thank you.
Excellent. Thank you so much, Sarah. Looks like we already have a hand raised. Sutapa, would you like to unmute yourself?
Yeah, I have unmuted myself. Can you hear me?
Yep.
It was a wonderful presentation. I really enjoyed it. And I have a couple of questions. One question was regarding, when you do the scanning and it gets scanned, how much time does it take for the whole scan? You showed one of them. How much time does it take?
Yeah, that's a great question. So it really depends on the qualifications that you're looking at.
Mm-hmm.
So if you have a higher resolution, say 500, and if you're looking at a larger scan rate, say 50 by 50 microns, and this scan rate is like 0.2 or 0.1, it could be pretty long. It could be maybe like a half an hour to 45 minutes. But there are times where if you do like 200 resolution with a smaller image and the scan rate is 0.5, it can be relatively quickly. It could be maybe like 10, 15 minutes, but it can be a meticulous process when analyzing a bunch of randomly selected locations.
Yeah, of course, I understand. And the other quick question was, regarding the AFM IR, and all, you are doing this in reflection mode or in transmission mode? I guess in reflection mode, right?
Absorb.
Reflective, like you're getting the sample signal off by reflection, or because in FTIR you can do transmission where it goes through the sample.
Yes, yeah, reflection. Yeah, it's also in a tapping mode.
A tapping mode. Okay, so the reason I was asking that is a lot of the particles, they have the environmental particles, they have a lot of stuff on them. So sometimes, yes, that becomes a problem. Thank you very much.
Yes, absolutely. Yes. Thank you for that question.
Presentation.
Thank you.
Shelley, you had your hand up next.
Yes, thank you for your excellent presentation, Sarah. I was curious, just, especially in context coming right after Harry's talk,
Yes.
when you take the 80 microliter drop of sample and scale it up to number of particles per liter, can we talk about the representativeness of the sample size and Was it just purely just numbers that you scaled up to that higher volume, or did you have just analyze how many samples, how many drops were analyzed to come up with that number?
Right, of course. Yeah, so it was the aliquot drop, and then in that drop, there were various subsamples of 50 micron samples. So I would say from my recollectance of those two sample drops, there were probably 20 or more of those locations. And then, of course, some of those locations, we did not identify any plastics, but some of them that we did. So using the counts of those plastics and then extrapolating to the 80 microliter drop, and then using that for nanograms per milliliter.
And what was the variability between stations or samples?
So I think I showed in the last or one of the conclusion slides is that the interesting thing is that we found a lot of polyester nanoparticles in the one, and then the other one we saw only PDMS. So meeting with the collaborators soon, we're going to do like more research and look at more wind patterns and see what the type of sources it could be. But right now we're still looking into the variance in them, because oh, sorry to interrupt, because looking at we have DI controls, and similarly, like the DI water goes through the same process that all the snow melt samples go through, and we run the infrared similarly at the same type of qualifications at 1735 and 1260, which was like a big peak in PDMS, and we haven't found any of that. We're trying to be very cautious about coarse contamination because we are looking at samples that are very, very tiny. But we still, till this day, are still continuing to do controls.
Thank you so much.
Yeah, absolutely.
I think we have time for one more quick question, Max.
Thank you. Thank you, Sarah. Max Zarate from the CDC. And I think your presentation is also showing us, the need for new methods in order to characterize micro nanoplastics physically and chemically. If not mistaken, AFMIR has that quality. You said that you can determine the 3 dimensions of particles plus identification of the chemicals. So, if not mistaken as well, I think your total mass was calculated using that tool, but you presented the total mass instead of like splitting PDMS and polystyrene. So what do you think are the applications of these two techniques with regards to the different types of matrices that we have in water?
Yeah, one of the complications that we're having right now is that snow water is relatively clean for the most part, and you really have to be sample dependent based on the environmental matrices. So in the future, if we were to look at ocean water, there's a lot of stuff in ocean water. There's salts, biological, there's organics. So one of the concerns that we have is that if we process it too much, if we digest it too much, that could be implicating the particles and the nanoplastics. And in addition to anything that's quite large a particle in the AFM. That's why it was nice to have the OPTIR to examine the larger particles, because what can happen when the AFM is in tapping mode, sometimes I describe it as like a needle on a record player. So when it taps, it records the height of the surface, but sometimes it can easily contaminate it. So we have to be very, very careful of when we're analyzing those samples.
Thank you. Good luck in finding a job as well.
Thank you so much.
Excellent. Thank you, Sarah, so much for that wonderful presentation. And I think we'll move on to the next speaker. So we're going to stay in this kind of nano size range, and we have Baizan Yan from Columbia University. Are you able to share your screen?
Let me try, yeah, just a minute. Can you see my screen?
Yep.
Okay. Let's
Three
see.
dots. Okay, yeah, perfect.
So great, thanks for the invitation. So yeah, my name is Beisang Yan. I'm a Lamont Research Professor here at Lamont Doherty Earth Observatory at Columbia University. So what I'm going to talk about today is nanoplastics and microplastics in pollution in the water, and as a continuation of Sarah's work, like showing that in her slides, which is tiny in the name but possible mega in impact. Let's see. So yeah, I'm just trying to give myself a little bit of introduction. So I'm first is an environmental scientist. So I'm doing the exposure, environmental sources and distribution and the behavior of the pollutants. Then I'm actually working with the medical schools and the Mailman School of Public Health to characterize the exposure and link the exposure to possible health outcomes. And then the third part of our focus is engineers. I try developing engineered solutions. For example, we have right now tried to developing the method for remove the microfibers from the laundry, and also try developing this like a smart sampling system that can collecting the plastics from the water. So what I'm going to go to talk about today is first to see the distribution of microplastic in New York City waterways, and then talk a little bit about my in situ experiments investigating the potential role of microplastic as vectors for transporting pollutants in Hudson River, and then the challenges studying the nanoparticles and our breakthroughs in quantify them in bottled water, and then potential, if I have time, probably we don't have time. If I have time, we'll talk a little bit about the engineering solutions. So I think that everyone already know this problem. I'm not going to go over it. So our first working study is actually about like 7 years ago when my colleague Joachim goes and he showed me this convincing picture that microbeads attached is actually in the South Korean Sea. You see that, like microbeads attached to the zooplanktons, the phytoplanktons. And also he has some of the pictures showing that actually the microbeads inside of the zooplanktons, and it was really, really convincing to me that our world was impacted by the microbeads and by microplastics. Then turns out this site was very close to the cosmical, manufacturers that basically use microbeads in there. So then I started to relate to collaborating with the riverkeeper Hudson, and then using their like a boat to get the samples from this is Hudson River, and also this is New York City, the Manhattan, and you can clearly see the microplastics. The concentration was increased when they are reaching to the like the waters in the like the close to the Manhattan. So also we can see our highest levels is actually Newtown Creek, which is a superfund size, which is filled with the plastics. However, in the New York City waters, the Hudson River was famous, was not for plastics, but it was actually famous for something else. For example, Hudson River is the longest superfund size because of PCB, pollutions. And so this is legacy pollutants. They are already in the environment. And so when this microplastic being discharged from the wastewater treatment plants, they're going to meet these legacy pollutants. They are going to carry them. So that's why we deployed these in situ experiments. And we did this one in the 2021. And we deployed this testing in 2 sites. One is Brooklyn Navy Yard, one is Newtown Creek. And in each site, we use these 2 stainless steel basketball. So basket, and in this stainless steel basket, we actually putting like a pellets inside. And these pellets, we first would drill a hole right in the middle of this pellet, which is about like 4 millimeters, in this 3 to 4 millimeters, the pellets. We dig a hole in there, and then we string the 50 pellets into this stainless steel wire, and then we attach to the 2 sides of the box. And we then, we go back to there for every a week, and then checking whether they are still there. And the second thing is we also basically brush this biofilm and also biofouling inside of the box. And then we this is whatever, 1 month later looks like, and then we took it back 100 days. And then we measure PCBs and the PBDEs, the bacteria, and also metals on the surface on these pellets. And so this is basically just showing you a result of the PCB because I don't have time to cover the other pollutants. And so we have 3 plots here. The first plot showing the concentrations of the PCBs in the waters from Newtown Creek and Navy Yard. And you can clearly see because the Newtown Creek was much more filthy than the Navy Yard, you can see the PCB concentration is higher. The red dot lines was red dots showing the concentrations was actually normalized to the weight of the particles, basically weight of the system particles. In here, you can see there was about 10 nanograms per grams of the suspended particles. And so the second plot showing the HDPE coming out from the virgin pellets and also the Newtown Creek and Navy Yard after 100 days of the in the field. And the concentration, this is the concentration of nanograms per grams pellets. And you can clearly see they are before that, the virgin one showing a much lower concentrations, and it clearly means that these PCB is coming out from the field, from the real environments. And then compared those with the concentrations in the suspended particles, you can clearly see there was even higher. And so it was about 10 times higher, clearly showing there was PCB enrichment to the particles, plastic particle on the surface. So, but consider the pellets it's much larger particles than the real suspended particles in the sediments, in the water. So if you normalize to the surface area, then this number will be about 1,000 times than the the concentrations if you normalize to the surface area. So clearly showing the extremely high enrichment for the PCBs to the surface of the pellets. And so one of my students can just mention that if you really have this plastics, HDPE plastic can really enrich the PCBs, you may use that for the remediations, which I really agree that we should explore that in the futures. And also that we also see the PVC and the pellets, and we clearly see the similar pattern. However, the enrichment of the PCB was not as high as the HDPE. We still don't know the reason, and we're going to do the more work in the future. So as I mentioned, I'm doing the health. To me, the health part is important, is the particles, the size is really matters. For example, why we study the PM 2.5 in the air is because when the particle size less than 2.5, they can really go down to the deep in the lung. And in the lung, then they have a chance to cross air and blood barrier and get into the blood. And through the blood, they can go to the vital organs. So in terms of the health, the particle size really matters, especially for these particles less than 2.5 microns. And so then this is, at the time when I look into, all the available method, I see some of the study that mentioned, used by Sarah mentioned, AFMIR, and some of the research on that. However, this instrument was pretty inexpensive and very expensive, and also, hard to have access to that. So that's why I try to see what's the regular method that we can use. And so you can use the SEM and TM, however, they are not really, specific. And so there was, I probably can identify the particles, but you don't know which plastic type is coming from. And also traditional Raman and traditional FTIR has the issues when the particle goes down to below 5 microns, and not even mentioned to below the 2.5 microns. And so this is actually a research, review paper in the, it's all, 4 years ago. And you can clearly see all the research showing that was identifying particles more than 23 microns. And so that's basically the situation we have during that time. So that I was really, because I really want to study the health. So that's really give me the the momentum to look into the various literatures, and not only including our, environmental science and the geology, but also looking into the chemistry and the physics and also instrumentation, field. And then I was coming across this new method called stimuli Raman scattering. And stimuli Raman scattering have two lasers, and then with these two lasers, then the sensitivity or the Raman signals was dramatically increased, about more than 10,000 times compared with traditional Raman. And because traditional Raman can goes to 5 microns, and since you actually increased all of magnitude in terms of sensitivity, then you based on that, you probably can go to the nano, into the particles into the nanometer. So I was so excited about that. So I contacted with looking into who is actually inventors of this technology. Find out one of the co-inventors was actually a professor of chemistry in our Columbia University. So that's why I was so excited and sending him the emails and talking to connecting him to see whether we can talk. And then half hour later, he said, let's talk. So then he's really looking for, for the applications using his, the tools that he invented. So then we got some of the intern money from the Columbia, and then we got, some of the money from the other places, and we starting to do this research. And we have two groups coming together. The first thing that we actually doing is basically try to figure out, whether these instruments can be used for identifying nanoparticles, nanoplastics. And this is basically showing that, we have this is about 130 nanometers, in terms of particle size. This is using polystyrene and as a standard, and then we can clearly see the particles and the intensity of the Raman signals was increased over until reach to the 1 micron size, etc. And so this is really giving a lot of the hope for us. So that's why we're so happy about that. And one thing that is actually even surprised to me is actually the Raman intensity. This is in the y-axis, and have a very good relationship with particle size in diameters. And you can clearly see this is a linear, a relationship, and until they reach to the 1 microns. So which means when we really see the Raman signals, we probably can figure out not only based on the laser movement of these particles of the spatial relations, but also we can based on the intensity, and then we can figure out their particle size, which is really it's an exciting thing for us. So after we set up this platform, so the first question is what kind of application we can use. So our group think probably bottled water would be the usage. The first thing is because they already have identification of the PET particles in the bottled waters, and so that's already been there. So the question is that do we have other particles? Do we also have the particles who goes to the nanoplastics levels, nano levels? So then we select the 3 popular brands, and then we did some work, and then we find out in this the average of that 3 brands, we found about 250,000 of nano and microplastics in the 1 liter of bottled water. And so so that's really about 2 order or 3 order magnitude compared with microplastics in the bottled water. And so we go, and then this method not only can see the concentration, but also they can see each individual particles, what's their distributions. And we find out, of course, there was a PET. And we know that that should be there because the bottle was made by the PET. You are supposed to see that. But also, we also see quite a lot of these others, like polyamide, PVC, and also polystyrene. So we are actually very surprised to see these results. The reason is because we know that in the bottled water, when they're making the bottled water, they use called a reverse osmosis filter. And a reverse osmosis is supposed to be remove the particle very efficiently. And any particles probably more than 0.1 nanometers, you're supposed to be removed by the reverse osmosis. And so why, after that particle being removed, why we still see the polyamide, polystyrene, PVCs? So it's really giving us the like the one thing we need to see is whether it's actually our method works or not. So we took it back and we basically look into more and also looking our results more carefully. After 3 or 4 months, we still get the same answer. So is almost 100% sure that it's actually polyamide, polystyrene, and poly PVCs in those particles, in the water. So question is where coming from? So, and then when we starting to writing this paper, we realized that reverse osmosis was actually use polyamide, use the PVC and the polystyrene make the reverse osmosis. So they are the materials made this, like the reverse osmosis. So it's ironically they can basically remove the particles from the source water. However, they are also shedding the particles into the water that be made. So that's, of course, this is still our assumption. And after this paper, the PNAS paper we published in this January, we got a lot of the media coverage, and also we got a lot of the, connections to see whether, especially industry, try to see our method. And then I asked the industry to see whether they can collaborate with us to let us to go to their sites and go to their collecting the water from each steps. And so far, I haven't got any feedback yet. So that's our kind of research in the bottled water. Now we actually think about what is actually in the tap water. So we are collecting the waters from across the United States. We are considering, this is currently we have, and also we are considering to get more into Wyoming into this inner side. And so, but we are already get some water from the California, but we get many was actually from the New York and New Jersey sites. So right now it's still working on this project. And so my students, and right now they are getting some of the preliminary results. And so preliminary results is quite a surprise. The reason is because when I got the interview from media, they always ask questions, is this tap water is more safe than bottled water? And actually at the time, I don't know. So I said, I don't know. So then now I look into tap water, and we find at least some of the city, some water from some cities, I find out they are quite, like abundant with certain plastics in there. So of course, this is just a preliminary work, and we still continue to do that. So then what question is, can we use this method for, for the, like biological samples, for example, claimed fish. And I use the rat samples as an example. So my colleagues, like Phoebe Stapleton from Rutgers, she exposed the polyamide to the rat, and then then after exposure, then she took the lung tissues and then makes this like a thin slide of the lung tissues, and our results showing after the exposure, you can clearly see there was a lot of these nanoparticles in the lung tissues compared with the control. So this method clearly can be used for like a direct measurement of the biological samples. But you cannot use that for the blood samples, because in the blood, the red blood cell was actually eat the plastics, and so they're basically like took the into their, and then our system cannot working because you have these bilayers of the fatty acids that which is preventing us to detect the inside. So we actually use the digest, and we digest the blood samples, and then we use the SRS. This is basically from 2 donors. One donors and the second donors are both from the areas which is close. And so we actually can see all the major plastics, like type, but I am only listing the, four like plastic, samples type, and polypropylene, polystyrene, PVC, PMMA, And we clearly see these two people was being exposed very differently. One people was exposed a little bit more in terms of the larger particles. One person was more into the small particles, into about 1 microns. And you can see we also identify the PMMA, which is, like a lot of them are used for the, like bottles, containers. And we can see they are very high level in the second person. And so they are based on our calculations and also density, we found is about 1 to 3 microgram per grams of the blood for the PMMA. So which is, like our current findings. And so I don't know how much time I still have, or I should continue, or what? Karen?
You have about 10 minutes left.
Oh, 10 minutes. Okay. So let's just talk a little bit about the engineered solution, which is, like we want to also focus. So basically, we know that in the environments, especially in the oceans, about like 1/3 was microfibers or microplastic was coming from the laundry. But it's in the environments of the, like New York City waterways, and this number can be reached to like 90% or even more. And so clearly the textile from the laundry was contributing to the, like our waters in the urban situations. And so the one thing is that coming out from the dryers, because they'll release that, into the air and then eventually deposited through the wet and dry deposition. And also washers, wastewater come from washers can get into the wastewater treatment plants, and then can 10%, based on some other study, showing the 10% goes to effluent and 90% get a sludge. And the sludge can be used for many things. And one thing is agriculture for the fertilizers, and then they are re-getting into the environment. So that's why it's a huge issue. So I'm actually using my home washers, and then I collected them, and then this is the result showing that putting onto the filter, you can clearly see a lot of these fibers coming out from the wastewater from dryers. And if I use that for if I use getting smaller samples, this is long fibers, but we can get like a more smaller filters, we can find out this micro plastic, which is in here using that ramen, we find out is polyesters. And then one big surprise to us is also this wastewater contains a huge amount of the microplastics in the nanoplastics in the wash water. And this number I haven't quantified because we are still quite, I try to quantify them, but the number is supposed to be more than like 100 times, even more than the microplastic being identified, clearly demonstrating this importance to study nanoplastics in this water. And so right now we are developing these filtration techniques. And so we got supporting from NOAA about like 1 year ago, and we are developing the filters. And right now this is a prototype, and we working out and to try to miniaturize it, and then to see whether we can incorporate that one into the water and the power supplies for the dryers and the washers in the like the new machines. So this is basically the research we are like doing. Another things we just got in news from NOAA that our another proposal we get selected was using the AI to precisely select and choose the plastic waste in the rivers and in the coastlines. And so basically you can see all these figures in here. For example, this is from the from the Bronx River. When they are using barrier, you can see there was a lot of the grass and the materials being mixed in there. And you can also see the plastic, but the majority of are actually the natural debris. And this is also showing the sea beans and also the collected from the other like collectors. And so when you're collecting these natural debris, first things there was no need, because they are supposed to belong to the natural systems, and which is good for the downstreams ecosystems. And so now you took that apart. And the second thing is it also add a lot of the burden to the maintenance. For example, for this company in here, and so they get a lot, but the only plastic was only 10% or less than 10%. So now they have to sorting that out. So which is get a lot of the burden, make it very difficult to be sustainable. So that's why we coming up with this idea to really developing this solar panel driving energy efficient and like a smart system that could be pick up this plastics and then only collecting these plastics, and also we can also monitoring the system as well. So this is basically what I my research, and I would hope that I will thanks everyone's time, and also this is my colleagues and my students, and also my funding source, and also a lot of supporting from the different organizations. Thanks.
Excellent. Thank you so much for that wonderful presentation. Really appreciate it. Anyone have any questions?
Maybe I runs too fast.
No, it was great. Really interesting. I'm very excited, and I'm sure this group will have to invite you back once we hear about you have some tap water results to share. Sutaba, would you like to start?
That was a great presentation. I really enjoyed it. I was wondering, you said that you can go down to a much lower size for detection. Typically, I think Raman, we have gone down to about a micron, but so the Raman that you are using, the results that you showed, they are 500 nanometers. That's about half a micron. Can you go? Is it possible to go down even lower?
Yeah. So that's a great question. So we, we can go down to currently the record is about 16 nanometers, and we can identify those particles very clearly. And so this is we can go down any numbers between the any number above that 16 nanometers. Yeah. So we can.
Yeah, go ahead. Sorry, I interrupted. I was going to ask you if this Raman is commercial, or is it just available within the lab itself that it is?
So yeah, so this is available in commercially. So but the problem is not really the instruments, is basically the time to spending and to training to get your technician people that are being able to running the instruments and to identify them. As you know, Sarah did a great job and to showing using the AFM-IR, but I think that that ability to identify them and then tuning the systems to make it happens, make it actually measure it is actually a lot of the training on that. So there was other labs also have that, but you need to really developing this kind of machine learning techniques that can identify them and also can quantify them. That is actually pretty much a huge amount of the basically effort to putting that into working on that. Yeah.
Oh, thank you very much. I really enjoyed your talk. Bye now.
Thank you. Thank you.
Excellent. Thank you. Brent?
Yeah, Dr. Yang, can you hear me?
Yes, yes.
I guess as the person who invited you to this forum, I want to express our appreciation. We're very grateful for your time. An excellent presentation. I was certainly not disappointed. I did have an actual question for you, though. As someone who also works in the membrane space, as you might imagine, your article caused quite a bit of consternation among the membrane manufacturers when they were reading about how the potential for microplastic shedding might exist. A couple of them I reached out to, and they had to do a deep dive with some of their scientists. They do not believe that the polyamide is coming from their membranes for various reasons, which I won't have the time to get into today. But I guess if you don't mind, I'd like to link you up with those folks to have a conversation about it. Now,
that'd be great.
That said, I said, okay, and the reasons that they articulated made sense. But I said, well, okay, where are they coming from then? And they didn't have a good answer for that. So there's clearly some more work to be done on both sides about how to figure out, at least for that aspect of your very good research.
So yes, thank you for Brent for the invitation, and also thank you if you can link me to the industry, because I tried, and I'm even goes to there was this UK the Bottled Water Association conference. They invited me to give a talk there. And I gave a talk there, and I basically in the slides, the final slides, I put in my information there. I said, I want to collaborate with you guys. Give me the samples. I can figure out each steps, what is really where it coming from. And so far was no, like 5 months later, I have no,
Yeah.
no response. I don't know what's the reason, you know.
I think the membrane manufacturers themselves would be a little more amenable to having that conversation.
I see that point. Yeah, but because as I mentioned, you're right. This is just our assumption. We don't really know where it's coming from. However, this is only assumptions because we know they are made by these materials. Like we know that the universal also, has the materials like polyamide or this PVC polystyrene. They are being used in the filter materials. But
Right.
we don't know. We don't really have the samples to direct demonstrate it. Of course, this is a problem, but can be really fixed if we can figure out that's really the source. Then we can use another layers of filter, maybe remove the nanoparticles, right? So I just don't know what is the reason leading to no response from the industry. I'm really keen to collaborating with them.
Yeah, I will certainly offline connect you, and we'll see if we can start a good conversation about that.
Thank you.
Thank you very much.
Mm-hmm.
Excellent. Max and then Sasha.
Sure, thank you. Thank you, Dr. Ian. A great presentation. And have a couple of questions. One, I don't know if you had any reaction from the bottled water industry. If you can share it with us. And second is, you mentioned, I think, that AFM IR is an expensive instrument for quantification and chemical identification of polymers, nanoplastics in this case. But if not mistaken, I think the platform that you developed, quite unique and strong instrument as well, is also an expensive instrument, correct? Are you working with regards to maybe make it more affordable to some institutions that may require to get your instrument? Congratulations again for your work.
Okay, so thank you for the excellent question. So the first question is the interaction with the industry, bottled water. As I mentioned, I tried, was not very successful. And I'm also trying to get water from desalination plants because the desalination use reverse osmosis. And they probably have also contributing to that, so I hope that in the future, I can get some industry collaborations to really solve the problem. And the second thing is also the second question you said is basically that how much compared with the AFM, IR. I don't know the AFM IR, but it looks like there are instruments to me. I think probably like more than like $3 million or something like that in that range. And so I'm not sure. I mean, Sarah may give a better idea. But the instruments we are using, we actually installed ourselves by my colleagues by myself. This about like $700 to $800,000. So it was much cheaper, and and also that can be like these instruments can be also used by many other researchers as well. So that's kind of things. As long as you have the instruments, you could potentially use many other things as well. So right now, what I'm trying to do is actually get supporting from the NIH to see whether they can purchase, an instrument for us that being installed in the Columbia University, specifically for the practical research work. Yeah.
I hope it happens. Thank you.
Thank you.
I had a question about how you might be, or if you're not subsampling or in order to get your concentration amounts, or also how are you managing if you have a sample with a large number of particles and distinguishing between particles if they're touching or not, some of those
Great
technical
point.
issues.
Yeah, great point.
And also thank you so much for your talk. It was very interesting.
So yeah, so thank you, Sasha. And this is, I think, as Sarah mentioned, so the natural samples contains quite a lot of this. I mean, she did the snow samples. I also did some snow samples from the Antarctica, and I would like to talk with her as well. So So, but the samples we got from, if we got from the like biological samples and also from environmental samples, it contains quite a lot of the other particles. For example, the, like the silt and also the, clay materials and natural, like natural organic matters, etc. So in terms of my research here, we actually developed a method that being, like basically to first to, to separate these nanoparticles and microparticles from those, like the other particles. So that's one thing we are doing so that we can basically shrink down the size and also can, allowing that, like the whatever concentrated is have a less of the interference from the other particles. So this is still, of course, in the for the biological samples, it's a little bit easy because there was not too much as natural environmental samples, but you still see quite a lot of the other, like we see the black carbon in the tissue as well. And so that can be interference as well. But so far was in the blood samples, we see actually a pretty nice signal that we can actually identify that. Did I answer your question?
Yeah, that was great. Thank you.
Okay, thanks.
Excellent, thank you. And there was a comment in the chat from Wynn Calgar. Wynn noted that there are naturally occurring polyamides that might be responsible for some false positives in IR. And what he means by that is when we use like a Fourier transform infrared spectroscopy, we do have to keep an eye on this and double check those matches. I don't know, Dr. Calgar, if you did want to expand on that, but I think Dr. Yan, I guess kind of building on that question is, yeah, how did you test for maybe things that may be false positives? Did you run, things that you may have found that could be a false positive? Like sometimes we'll, like Sasha recently was telling me she was grinding up pine needles and checking to make sure that they weren't an interference. Did you do anything like that in your study?
Yeah, certainly. This is a really great questions, and so that's that's what our lab try to do. That in the natural in the bottled water we don't have that problem because we directly filtered the samples. We basically pour the samples to the filters, so we didn't really touching any environmental samples and also our lab samples. We find in the lab. In our lab water, like a molecule water, we see a lot of the plastics there. So it's clearly, that's a, like a contaminations as well. So our currently our method is to remove basically try, of course, this is my hope, my dreams, is that the Columbia can get some of the, goods, like donors that can potentially help me to set up this plastic-free lab so that I can remove the particles, remove the plastic from the air, from the water, from in our lab environments. That can really answer your question, Wayne, that like how we can minimize this background. However, our research also showing that we also have a very strict QA/QC. So basically, we have when almost every samples we have this, like a blank and procedure blank samples that allowing us to tease apart the really contamination coming from the samples and from the real samples. That's whatever we can do right now. But it's in the future we certainly need to get the more resource to set up this lab so that they can be able to do the things more clearly and more firmly. Yeah.
Excellent. Wynn, did you have anything you wanted to add?
No, I think that's great. Thanks for answering that. I'm not sure about the false positives specifically with ramen. I just know it's a common thing that we're dealing with right now with IR. Lots of different false positives and false negatives in spectroscopy that need to be squashed, but you're working with a very new method. So I'm sure there will be a lot of research to come to figure out all these details. So thank you.
I completely agree with you. I mean, not only in our field, but also in the pyrolysis GCMS field, was also I can see there was potential, like a false positive. However, as I mentioned in our study in the bottled waters, I don't think we have that issues because we never touch any water and any air during our sampling processing processes. And we have a very strict QA/QCs. Yeah.
Excellent. Thank you so much. I am concerned that we're nearing the end and I still see some hands raised. So Dr. Yan, would you mind putting your email in the chat, and those questions that we maybe don't get to right now, they can email you their questions. And I just wanted to leave maybe the last few minutes to see if there were any general announcements for the community, any events that are happening or anything. Anyone have anything they want to share while we're all here? I'm not seeing anything.
Monica, I would mention that, you know, we're having meetings about quarterly, so we'll send out a next meeting date. And if you have any ideas about speakers, people you want to hear, please let us know. We're well connected and can get speakers usually. So if there's any particular topics you're interested in, let us know. Also, you know, we have the subgroups for communication and for the playbook. The communication one looks like it is finished. So if there's anything else that you would be willing to help lead and something you want to focus on, please feel free to propose another subgroup or another topic for a subgroup. We'd appreciate that.
Okay. With that, shall we end the meeting?
Sounds good. 2 minutes early.
Yeah, 2 minutes early.
Great talks, you guys. Thank you.
Thank
Thanks,
you.
everybody.
Thanks
Have a
for
great day.
coming.
Thank you.
Bye-bye.